So, welcome everyone to our October webinar for the CGC. Today we're going to talk about 3D Slicer and its usage in the cloud. Our speaker today is Dave Robertson. He's a community engagement manager at Velsera. You've probably spoken to him in office hours. He solves everybody's problems. He is a specialist when it comes to scalable, reproducible analyses on the cloud. He's passionate about helping researchers maximize the value of cloud computation technologies. And he's here to talk about 3D Slicer. So whenever you're ready, take it away, Dave.
Thank you for that fantastic introduction. Just give me 1 second to share my screen and I'll be ready to go. Okay, can you see that coming through?
Looks good.
Fantastic. Like I said, thank you for the fantastic introduction, everyone. I'm Dave, and today I'm really excited to be here to speak to you about how we're enabling 3D medical imaging for cancer research on the Cancer Genomics Cloud. So over the next several minutes, I'll explore the powerful new capability that brings advanced imaging analysis directly into your research workflows. So what are we going to talk about? So this is what we're going to cover today. And I know this is an image of a heart, but I thought it was really fantastic because it's really showing the 3D nature of what's possible with this software. But we're going to have a cancer-specific use case in the demo. So first, we'll talk about why 3D imaging is so critical for cancer research. Then I'll show you the technical solution, which is called Slicer Jupyter. And we'll show you how to run it. We'll do a live demonstration, and then we'll explore some research applications. And then, if there's time there'll be plenty of time for questions if there aren't any. Okay, so why does 3D imaging matter today? I think it's because of three reasons. First, cancer research increasingly relies on these complex imaging data. So you have computed tomography, MRI, and PET scans. And we simply need researchers like you need those sophisticated tools to analyze them. Second, cloud-based solutions remove computational barriers. So you no longer need expensive local hardware. I remember in my laboratory, our imaging specialists had their own workstation, like specialized workstation under their desk that would do the imaging. So that's no longer needed. Because you have the Cancer Genomics Cloud to help you. And finally, you can integrate these imaging data points into your current CGC workflow. So you can not only do multi-omics, but you can do multimodal. And we'll talk about that a bit more. So what can you actually do with these things? Well, I want to break it down into two major areas. You can have advanced clinical insights and more research power. So I know CGC is generally for research, so I just want to caution everyone there, but you can sort of validate certain things. So on the clinical side, you could measure tumor volume. You could track treatment response over time and plan additional precision medicine therapies. For research power, you can extract radiomic features, train AI or machine learning models, and you can scale up using batch processing. So how did we implement this on the CGC? So Slicer is implemented through Slicer Jupyter. So this essentially gives you a full JupyterLab environment in the cloud. So I just want to take a second to talk about this. So a lot of researchers may have more experience thinking about 3D Slicer as a desktop environment, which it is. But we implemented it as Slicer Jupyter because, one, it was the fastest way to get this going for the CGC users. Two, it actually adds a lot of benefits you may not have thought about. First, because it's inside of Jupyter, it actually is running Python. So all the Python packages are available. So you have Python scripting fully enabled. Of course, because it's on Data Studio, it joins a family of other Data Studio environments and that are super popular. Of course, JupyterLab, RStudio, Galaxy, OHIP Viewer, SAS Studio, and the newcomer Slicer Jupyter. There's no installation. You just launch it, and then you can customize if you want graphical processing units, otherwise known as GPUs, and how much attached storage, and of course, do the suspend time and everything. So you'll have direct access to the CGC data, either the bring your own data or the other data nodes that connect directly to the NCI's Cancer Genomics Cloud. So there's no more downloading, there's no more moving files around. So let's just talk about a typical workflow. So if we zoom out, a typical workflow is essentially like you start Slicer Jupyter, you load up your images, you can visualize the images in 2D, and then you could visualize them in 3D, segment them, do some measurements, and export. And then when you export, that's when you could essentially move the data back into your CGC project and combine it with another data set. So in this case, I'm thinking of, you might use some of the measurements as a phenotype for genome-wide association study, or you might combine some of these images with a single-cell RNA-seq analysis. So let's see this in action. So for the live demo, I'm going to go to the CGC platform. I'm already logged in. But what we're going to do is we're going to talk about loading cancer imaging data. We're going to create a 3D visualization. I'll show you how to use the notebooks. Then we'll run a automated tumor segmentation and extract some quantitative metrics. Okay, so I'm already logged in. Actually, I'm in the web browser right now. So I'm just going to move one tab over, and I'll show you, this is what the typical CGC interface looks like. So of course you have your projects on the left side, and you have your most recent analyses on the right side. Today I made a new Slicer 3D webinar project, which I'll open up here. And of course you can do the typical membership that we show you a lot, because it's all about collaboration. So let me just add my colleague Rowan here, just to remind everyone that you can share. You can share your analyses. So what we're going to focus on is just one tab, which is the Data Studio tab, because as I said, Slicer Jupyter is part of the Data Studio suite of tools. But let me just show you how you create a new one. So if you create a new analysis, you get to pick which one. So we're going to, and you can have more than one running. So you could have, more than one project being analyzed at the same time. And you can also share things and segment things by which collaborators you're working with. So I'm just going to call this 3D Slicer Part 2. And then there's only one environment, but in the future, we may have additional custom features. And then, of course, here is where you would pick. The default is the C5.2, which I'll keep it at. It's only 34 cents per hour. But if you do want to scale up, we should have in this list some more modern GPUs. So the G5s and I believe the G6s are going to be what you want to look at, and they actually, that's a pretty large one. Let's take a look at G5 16 or G5.8. G5.8. So at $2 per hour, that's pretty good value. So you'll only be using this if you need to train or run a machine learning model. I'm not actually going to start it, but then you would click start and wait 1 or 2 minutes for it to load up. So I already have this one running. So I can just click open, and it'll open up in a new tab. And it essentially is your typical JupyterLab environment if you're used to seeing it. So one thing you'll notice that's different is you have this slicer icon right here. So it's open source software, but it essentially gives you all the features of 3D Slicer, but available inside of JupyterLab. you can also access it from the console, and then you have all these other features. So if you did want to open a terminal and install additional packages, you could as well. What I'm going to walk us through is some of the examples. So these are, every time you launch 3D Slicer, these will be available for you. So if you forget how to do something, feel free to look at these. And there's 4 main notebooks that go from data loading, processing, additional image processing, and segmentation. So we're going to just take a look at quickly all 4 of these. Okay, so I removed all the outputs, but typically when you start it, they're going to have some outputs there for you. I just want to walk you through what's available. So, and by the way, on the left side here, there's a data folder. So it's pretty much just using 2 data types. So, and the main one we're going to focus on is MRI for brain tumor. So it's already sort of been ingested. Typically, you would get the data in sort of a raw DICOM image, and there will be a whole bunch of them. So this actually is 1 step further down the loading part. So it's actually collecting them all into 1 file called an RRD file. And so I'm just going to go through some cells. So by the way, if you're not used to using Jupyter, it goes through the codes run in a code cell, but then you can also add additional cells. My favorite keyboard shortcut is Escape and B. So B is going to add a whole bunch of cells below wherever you are, and you can simply just type in text. So it's a great way to take notes. Okay, so I'm just going to run some of the cells. So that was a setup, and then here's where it's actually going to load the NRRD file, and you'll see the 3, it's basically 256 images. And this will show you. Now with the MRIs, they're actually from 3 different angles. And so I forget the terms right now, but they're from 3 different angles, and it'll be really clear in a second. Oops. Okay, so here's just all from the same angle, and sometimes it refers to them as colors. So that's why you see this is the red angle, and so this is a tumor. So you'll start to see as we progress through the tissue slices, you'll start to see the tumor pop up here. Okay, and here's the 3 angles. So and as well as a rendering of this is called a 4-up view. So okay, so that's great, but we actually want some more interactivity. So this is going to show you some more interactivity. Here's a great way to do it. So this will actually allow you, using the same angle, the same pane, be able to go through the body into where the tumor is, and you'll be able to see that from the 1 angle. Okay, so here's another way to show you really the dynamic ability of the Jupyter Notebook. So this is a lot of the features that you would get on the desktop version. So essentially, you could zoom in and zoom out to see the tumor. So this is more of the visualization, 3D visualization and exploration view. Okay, and then we have the 3 panes here. So this is loading up an interactive widget that you'll be able to interact with via a mouse. So while that is loading up, I want to open another example, which is, I believe, this one. Okay, so this is using different types of filtering. Oh, we have someone to join us. Okay, yeah, so this is where I want to sort of get. So this is using a different data set, but it's important because it's actually showing you the chest cavity. And what you can actually do is, so this is doing it all in real time. That's why it's taking a little bit, but it's actually going to show us a 3D rendering of the chest cavity. It should show us the chest cavity and also essentially 2 points in the chest cavity. So if you had, if you wanted to find not only the tumor volume or the distance, but you have the distance of 2 points in the chest cavity. Let's see if this actually loads it up. Okay. So this is save the model. Let me see. Let me just bear with me as I. Yes. Okay. So once again, we have the what's called the 4-up view, which is the face. And by the way, so this is essentially the data that just comes with installation. But there is software if you're concerned about the face reconstruction, you could blur that out if you're using your own data. And then here's the last notebook here. And this will walk you through essentially segmentation. This one may actually, oh, no, okay. So here it is. So it's actually able to segment where the tumor is in this particular angle. Okay. So any questions about that so far?
I have a question, Dave.
Sure.
I just want your confirmation. All of these notebooks are preloaded, right? So you don't have to come up with your own code or learn something from scratch.
Exactly. And so in fact, they will be there when no matter what project you're in, you just start JupyterLab and you'll see those as in the example folder. So it'll probably start you in this folder and you'll always have the examples to go back to. In fact, that might be a good way to learn how to use it. You can just copy these and replace the part where it loads it, which I think is this cell. Oh, so it's just doing, it's just loading sample data here. So we'd essentially change this and point it to your actual data set, which could actually be in your CGC, in the CGC project. So it wouldn't have to be here in the data folder. You could, which I actually would advise against, especially if you have thousands of DICOM images. I would keep those in your CGC project and just point this code box, this code cell back to your CGC project. Okay. So that was sort of just like a quick overview of some of the example data sets. And I just want to wrap up with a couple more slides. So it's quite powerful. I only looked at some of the example notebooks, some of the surface features. But let's talk about like what are the research applications. So being the CGC, the most important, or the most like critical would be tumor analysis. So volume measurement, like I said, I didn't get a chance to show you, but it's in part of the examples. You can actually get two different locations, maybe two different tumors, and measure the distance between them. RESIST criteria. So RESIST stands for Response Evaluation Criteria in Solid Tumors, which is a standardized set of rules to evaluate how the clinical treatment's working. So you could essentially like develop automation around looking at the RESIST criteria, radiomics and AI. So of course, extracting features and training machine learning models, treatment planning. This kind of gets more into the clinical aspect, but we have clinicians that do use the CGC for research purposes. So if they're planning out protocols or making new sort of scripts, that would be very useful. And of course, ultimately, multimodal. So PET and CT fusion and imaging and genomics. We're seeing a lot of excitement around that. So what are some of the key benefits? Rowan already mentioned this, but reproducibility is huge. So versus, now remember, we're using Slicer Jupyter as opposed to the desktop version. And the Slicer Jupyter adds additional sort of built-in aspect of the notebook. So you'll be able to take additional notes and run your code. You could potentially also do version controls with GitHub. Of course, automation. We can do batch processing very, very easily on the CGC. So if you did need to scale up, you could essentially export exactly the same environment, the Slicer notebook environment, but run it as a CWL app, for example, or Nextflow app. Of course, the example notebooks are fantastic for education, and they make it easier to train new students or new team members. And I already mentioned the cloud power. So to wrap up, I just want to say that we do host office hours, and we love when people come. So they're twice a week, 10 a.m. on Eastern time on Tuesdays and 2 p.m. Eastern time on Thursdays. And of course, if you need any additional help outside those hours, I know not everyone is available then, feel free to email support@velsera.com, and they can get you in touch with us directly, or if it's a question they can answer, they'll answer it pretty quickly. So thank you for your time.
That's fantastic, Dave. While we wait to see if we have any questions, yeah, I just wanted to say a few words. So as we wrap up, like I said, as we're waiting, we hope you've seen just how powerful 3D Slicer can be. It's such a good resource for imaging data. You can load, visualize, analyze everything, as you said, Dave, from MRI to microscopy in 2D, 3D, even in 4D. So now with those capabilities being directly in the browser, you can combine them with a lot of different resources, like connecting them directly to your CGC project, as well as accessing data from various resources. So speaking of, we also encourage you to explore the NCI's Imaging Data Commons, the IDC. This is a cloud-based hub of cancer imaging data. It's co-located with analysis tools like 3D Slicer, and it blends very well with the Cancer Genomics Cloud. So IDC provides open, standardized access to diverse imaging data sets. It helps researchers connect imaging data with other molecular and potentially clinical data across all of the Cancer Research Data Commons, or the CRDC as it's called. So whether you're developing new methods or you're training models or trying to just visualize something, keep the IDC and Slicer, Jupyter, 3D Slicer, any of that in mind, because they really work well together to form a powerful foundation for imaging-based cancer research. So if you have questions, like Dave said, visit us in office hours. We're happy to just share what we know with you and help you find resources that'll get you going. Email us, whatever it takes. We want you to be successful. So thank you.